Welcome to the CJD Foundation virtual conference and today's session, an overview of prion protein monoclonal antibody PRN100 therapy for Creutzfeldt-Jakob disease. We thank our partners, the Centers for Disease Control and Prevention, and the National Prion Disease Pathology Surveillance Center for helping make the CJD Foundation virtual conference possible. We also thank all who have donated to the CJD Foundation or through Strides for CJD. Your gifts help support all of the Foundation's programs: family support, medical education, research grants, and conference presentations like the one you'll see today. We're pleased to welcome Professor John Collinge, who is Director of the UK Medical Research Council's Prion Unit. Please enter your questions as they occur to you during the presentation. At the conclusion of the session, we'll address as many questions as time permits, focusing on tonight's topics.
It's my pleasure to introduce Professor John Collinge today, speaking to us. He graduated in medicine from Bristol University, trained in clinical neurology at St. Mary's Hospital and the National Hospital for Neurology and Neurosurgery in London. He's currently Professor of Neurology at University College London, Institute of Neurology. He's also Director of the UK Medical Research Council's Prion Unit, a highly multidisciplinary research unit focusing on human prion diseases, and leads the National Health Service National Prion Clinic at the National Hospital for Neurology and Neurosurgery, where he is an honorary consultant neurologist. Professor Collinge is a true leader of the field. He has done much for our field and continues to further it. And it's my pleasure to introduce him to talk about PRN100.
Okay, well, I'd like to start by thanking Debbie and the Foundation for the invitation to talk to you today. I'm going to talk about a treatment program we've recently completed with a monoclonal antibody in Creutzfeldt-Jakob disease. Firstly, a financial disclosure. I'm a director and shareholder of a spin-out company which manages intellectual property that arises from my unit, including on PRN100. So prions are lethal pathogens, which are biologically unique. They cause fatal brain diseases, including CJD in humans, scrapie in sheep, BSE in cattle, and chronic wasting disease in deer and other species. They're different from all other infectious diseases caused by bacteria or viruses, for example, because those have their own genetic material, enabling them to make copies of themselves. But prions are just composed of protein and have no genetic material of their own, and they're formed from a misshapen form of a protein known as the prion protein. Now, the prion protein is one of the body's own proteins, so our natural immune defenses do not work normally against them. We don't make antibodies against them. And that's one of the reasons why prions are so lethal as infectious agents. Now, the normal prion protein, shown in this cartoon here, sits on the surface of our brain cells. It's a normal brain protein. But in the disease state, what's happening is that this protein unfolds and forms these misfolded chains, which stack against each other, forming these long fibers. And it's these long fibers of this misfolded protein that constitute the prions. And they propagate because if you start off with a small prion, a seed as we call it here, it grows as more and more normal prion protein molecules unfold and stick to the ends of it. And as these fibers grow, they break up, they're fragile, and they split in half, and then you have two seeds. They grow and then they split. And so you essentially have a chain reaction which results in more and more of these fibers growing in the brain. And this process eventually causes death of brain cells and the onset of the neurological disease. Now, there isn't simply one abnormal way these proteins can fold. There are multiple different abnormal shapes, and these constitute the different strains of prions, just as viruses can come in different strains. So you can have different strains of prions, and they produce different patterns of disease in patients. And this is important as we come on to talk about our therapeutic strategy in a moment. So the MRC Prion Unit in London was set up at the end of the 1990s at the request of UK government. And one of the things we were tasked with as is trying to develop effective treatments for these diseases. The key thing is, what's the best target for a new drug? What is it we want the drug to actually attack? How do we validate that target? How do we ensure it's the right target? How do we measure whether a drug actually works? That I'll come on and talk about. And principally today, of course, I'm going to talk about our development of therapeutic antibodies as a treatment for CJD. Now, as I've mentioned, prion replication involves recruitment of normal cellular prion protein, referred to as PRPC, into chains of misshapen PRP that form prions. So if there's no PRPC there, if you remove the PRPC, prion replication logically simply can't occur. So this is a very good target, we think. However, before targeting PRPC in the brain, we want to be sure that degeneration of the brain is not caused by losing the normal function of the protein, which of course is a possibility. And we started off by looking at that quite some years ago. Giovanni Malucci, then working in the laboratory, managed using a genetic method to switch off prion protein expression in adult brain. And fortunately, that didn't do any harm to the mice. So we could rule out that loss of the normal function was a sufficient cause of degeneration of the brain. And then she was able to go on and switch off prion protein expression in the brain. The technical details of how we did that don't really matter. But she found that when she switched off the protein expression in the brain during the actual brain infection, the mice didn't develop the disease at all. They were completely prevented from developing the disease. We were delighted to see that also the early pathology, the early damage in the brain, you can see here down the microscope, was actually reversible and improved. And similarly, behavioral changes in the mice, behavioral abnormalities in the mice also reversed when the prion protein expression in brain cells was switched off. So that was very encouraging, and we felt validated the cellular prion protein as a target. We could target it in adult mouse brain without major consequence. We can completely block development of clinical disease in mice with established brain infection. And the early pathology in the brain and behavioral problems, at least in mice, are reversible at the early stages. So, as I've mentioned, the cellular prion protein needs to unfold and radically change its shape to adopt the form that's seen in the prions. So, any molecule that binds tightly to the normal folded form of PRPC should prevent that process and should help block prions propagating. Now, we and other colleagues around the world have tried for some years to develop a small molecule, a sort of tablet drug, which binds to the prion protein and would do that. It's proved extremely difficult. So antibodies are molecules naturally produced by our immune system, and they've evolved to very tightly bind to proteins. That's what they do. So this is the approach that we followed. And Charles Wiseman, working in the unit in the early 2000s, showed that you can cure prion-infected cells in tissue culture dishes in the laboratory using anti-prion antibodies, which was very encouraging start. Now, I mentioned that prions come in different strains. However, strains aren't completely uniform. If you look at a brain here, these symbols are meant, the blue symbols are meant to illustrate the predominant strain that's infecting that brain, but there are also minor components showed by these different colored shades. The strain isn't all like, the prions aren't all identical in their shape. Consequently, if you develop a drug, a different strategy now, where you develop drugs that bind to that abnormal form of the prion protein, it may be very effective in getting rid of the dominant component shown here in blue. But what will then happen is the minor components to which the drug doesn't bind efficiently will then take over, and you'll have a resistant infection. Many of you will be familiar, of course, with drug resistance to conventional antibiotics. And indeed, Dr. Prusner in San Francisco, developing drugs like this, did find this to be a problem. The strategy we're using here, antibodies binding to the normal protein, not the rogue or disease form of the protein, should work against any strain. Because all the strains require the normal prion protein in order to propagate. So in principle, we ought to be able to cure the infection. So in the early days of the unit, in the early 2000s, we made a whole load of antibodies against the human prion protein by injecting the human prion protein into genetically modified mice, which didn't have the protein, and so their immune systems hadn't seen it before. And of those 100 or so antibodies, we came out with what we thought was the best candidate antibody, one called ICSM-18. And we went on to show in studies with David Anstey and Simon Hawke, published this in Nature in the early 2000s, that monoclonal antibodies could inhibit prion replication and delay the development of prion disease in mice. So that was very encouraging. The Medical Research Council in the UK then funded us to make a human version of ICSM-18. You can't treat humans with a mouse antibody. Your immune system would recognize it to be a foreign protein and would destroy it. But using genetic engineering, it's possible to engineer the mouse antibody until it becomes a human antibody. And we make a particular type of this antibody. The technical details don't matter. It's a G4 type. And the reason we do that is antibodies, in addition to sticking tightly to their target, the other end of the antibody can activate the immune system in different ways. And that's not what we want. We simply want this antibody to stick to the protein and prevent it being recruited into prions. We don't want to cause inflammation in the brain. And the G4 isotype has much less of that immune function. So that's why we engineered it in that way. This is a binding curve. The technical details don't matter, but it simply shows that the human antibody we made is really indistinguishable in its binding characteristics, the human prion protein, to its mouse equivalent. And we showed also that it has similar properties in terms of cell curing and in mouse models. And we then took this antibody through the normal preclinical safety testing that's required before one could consider administering a drug to humans. So we'd achieved what's referred to as a proof of principle for prion immunotherapeutics, for treatment of prion disease with an antibody. This seems to be plausible. But there are two key issues we've got to deal with here. One is the so-called blood-brain barrier. This is a natural barrier which protects our brains from substances coming into the brain which might disturb its function. And the brain's particularly good at keeping antibodies out, this barrier. So we have the question of, will enough, if we give the drug by intravenous infusion, which is the way one would normally give antibody drugs, will enough of it actually get into the brain? Now, an alternative way of delivering it is by infusing the antibody directly into the fluid-filled spaces of the brain. But that requires a neurosurgical procedure to put a tube into those spaces, and we wanted to avoid that if we could. But that was certainly something we considered to do if it was necessary. And then the second big issue is, will the dose be limited by side effects? All drugs have side effects. It's if you give sufficient dose of them. The question is, can we get sufficient of this drug into the brain to treat the disease without it being limited by side effects? So those are the two big questions. Now, I'll just say a little bit about how we measured change in the patients. In the UK since 2008, we've been doing something called the National Prion Monitoring Cohort and recruiting many patients for long-term studies of the natural history of prion disease. We've recruited more than 1,000 individuals to that study now over that period. And one of the things that allowed us to do was to develop a new rating scale to monitor progression of the disease. This we published some years ago and is in wide use around the world. It's a 20-point scale. 20 is normal, and it measures various functional activities in patients. Now, I should say that there are two normal different forms of the prion protein. All proteins are composed of a string of amino acids, building blocks of the proteins. The human prion protein has 253 of those. And at position 129, there's a variation. You can either have the amino acid M or the amino acid V. And so everybody has a different genetic type, which could either be MV, MM, or VV, depending whether you inherited two different forms from your two different parents or two of the same type from your parents. Now, MV, which is the commonest type, and about half the people listening to this presentation will be MV, gives you a reasonable degree of protection against developing prion disease. And if you do get a prion disease, it tends to progress more slowly. And that's relevant, of course, when we're monitoring progression of treatments in patients. This is work Simon Mead in the unit published several years ago, showing what's essentially the MRC scale score here. And as you can see, patients with the MM form tend to progress very rapidly with the disease. The MV form much more slowly on average, and the VVs are in between. And this is quite important when one's monitoring progression of the disease. So we published our experience of treating patients with PRN100 in the journal Lancet Neurology in April this year. And we arranged for that to be an open access article. So this is freely available to anyone around the world. You can Google this and download the full paper. And there's a large supplementary appendix of about 40 pages, which is a great deal of detail about how we made the antibody and tested it and the individual clinical progression of the patients. So this was the first time this antibody had been used in humans. So obviously we proceeded cautiously, and the patients all had rapidly progressive forms of CJD, predominantly sporadic CJD. This was conducted not as a regulated clinical trial, but under what's called a specials license with governance from a hospital oversight committee. And the plan, which was approved by the Oversight Committee, was for very cautious intravenous dose escalation. We started with a very low dose. All the data and any evidence of side effects was reviewed by an independent subgroup of the Oversight Committee, and they approved us to proceed to the next dose in each patient, up to the treatment dose of 80 milligrams per kilogram. And we then plan to give that dose every 2 weeks thereafter. And we assessed the patients using the MRC scale I've just mentioned and measured concentrations of the drug in the blood and the spinal fluid throughout. Now, as I mentioned, we had an option to go to administer the drug directly into the fluid-filled spaces of the brain if that was necessary. It turned out it wasn't, as I'll come on to show you in a moment, thankfully. So the first patient was treated just at the end of 2018. Patients that lacked mental capacity under English law, there has to be a decision of the Court of Protection as to whether the treatment's in the patient's best interests, and the court judged that it was, and 6 patients in total were treated. I'm pleased to say we saw no acute side effects and no evidence of side effects after treatment over several months in some of the patients. So that was very positive that we didn't see any evidence of toxicity. There was no need to progress to intracerebroventricular treatment. That's giving it directly into the fluid-filled spaces of the brain. As I'll show you, the CSF levels were encouraging. Now, I should say that because we treated inevitably a very small number of patients here, because we only had a finite small batch of the drug, and because the disease itself is very variable from patient to patient, the only way we were likely to show clear clinical evidence of efficacy is if one or more patients halted in their decline, they completely stabilized, or even got better. We've never seen that before in our clinical experience in the national cohort. So if that happened, I think we could interpret that as the drug working. If that didn't happen, then it's very difficult with a small number of patients in a very variable disease to conclude much clinically about whether the drug was working or not. So these are showing you the levels of the drug in the cerebrospinal fluid. Our target therapeutic concentration was 50 nanomolar, and you can see in the 4 patients that were on the drug for long enough, they all reached that target, and indeed some cases considerably exceeded it. So that was very positive. Now, these are the trajectories on using that MRC rating scale for each of the 6 patients. There's a lot of information here, and I don't have time in this short talk to go through in great detail, but all this is available in the papers that I mentioned you can download. In red, you see the individual patients that were treated, and in the background there, you see lots of other patients who were recruited in the National Prion Monitoring Cohort and are matched by genetic type and also by starting at roughly the same point on the MRC scale. Now, although, as I say, we can't conclude clinical effect from this small data set, there were some encouraging signs in patient 1 while they were on, I should say, when this line goes green, each red arrow indicates when one of the therapeutic doses was given. When it goes green, when we pass that threshold of 50 nanomolar in the cerebrospinal fluid. During this period, this patient was treated, we did see leveling off of the decline Also in this patient had iatrogenic CJD. We compared in this case against all trajectories we had for that subtype of Prion disease. You can see this patient lived longer than we've seen before with any case of iatrogenic CJD. And again, we'd like to consider that there was some leveling off on drug. I mean, that may be wishful thinking on our part, but we did find this encouraging. Similarly with patient 4, who had a very rapid decline to begin with, but when they reached the therapeutic levels in the spinal fluid, it did seem to stabilize. Unfortunately, by this point, with patients 4 and 6, we ran out of drug product, and obviously we could no longer continue the treatment. These 2 patients with the MM form progressed very rapidly and never reached the therapeutic level in the CSF. Now, patient 2 kindly consented to autopsy, and we were able to look at the brain tissue down the microscope. And this turned out to be extremely valuable information. And here is a patient who had not been treated with drug. We looked at many of those. And this brown staining here is where the prions are. This is the abnormal prion protein stained looking down the microscope. And you can see here in the patient that had been treated with PRN100, this is pretty much washed out. There's much lower levels here. We found this very encouraging indeed. It indicated that our drug was indeed getting into the brain and engaging with the PRP target and beginning to clear it from the brain. And furthermore, we also found that there was deposition of this abnormal prion protein around blood vessels in the brain and in the membrane surrounding the brain. You don't see this in untreated patients. So again, it was suggesting that the drug is helping the prions be cleared from the brain. So we found this very encouraging indeed. So to summarize our study, this was an academic-led project which generated a humanized anti-PRP monoclonal antibody and oversaw manufacture to industry standard for human use. With governance oversight from our hospital trust, we treated 6 patients with CJD under a special needs license. Patients were treated with increasing doses of PRN100 by intravenous infusion and closely monitored. Intravenous administration was safe and able to target the brain compartment, and no clinically significant adverse reactions were seen. Now, while in no patient was disease progression halted or reversed, in 3 patients, the clinical rating scores did appear to stabilize for periods when CSF drug levels were in the target range, but the small numbers precluded meaningful statistical analysis. Neuropathology of a patient with the longest clinical duration we've yet documented in iatrogenic CJD showed no cytotoxicity, but did show a marked drug effect with modification of deposition of disease-associated PRP. We went on to actually measure, we were able to measure actually the brain tissue-bound drug levels, not just what was in the spinal fluid, but actually what was bound to the brain tissue itself. This is what really matters. And we found levels equivalent to between 66 and 182 nanomolar. So well above our therapeutic target was reached in tissue. And I should say, in cell experiments in the laboratory, we find of the order of 1 nanomolar is effective in treating cells. So we're getting into a concentration that should indeed be efficiently blocking prion propagation. Now, we only had a single batch of PRN100 available, so it could only be offered to a small number of patients with well-established neurological disease until the drug was exhausted. And the fact that we proceeded with a cautious dose escalation, because obviously it was the first time this drug had been given to humans, it meant it took a highly significant time, on average 47 days, to reach our target level. Now, as many of you will know, CJD is, of course, a very rapidly progressive disease, and 47 days is a very long time. Indeed, it may be the remaining lifespan for some patients with established CJD. So that was too long. And obviously, in future studies, we'd want to, now that we know that it's safe, would ramp up the dose that we'd hope to get this sort of concentrations in 48 hours rather than 47 days. We've established a safety profile encouraging CSF and brain levels following IV administration, and preliminary clinical data, in our view, justify now a formal efficacy trial. And we want to look at patients in the earliest possible clinical stage before extensive neuronal loss is present and irreversible secondary neurodegenerative processes are established. Because what we think is going on when patients have well-established disease is that, of course, brain cells are dying. And as brain cells die, they themselves release toxic material that will damage cells around them. And that process itself won't be stopped by blocking prion propagation. In a sense, it's too late. So we have to get there earlier, where blocking prion propagation before extensive cell death has occurred. And of course, as I mentioned, we want much more rapid dose escalation. Now we know how long it takes to reach those levels. If those studies were positive and we had clear evidence of clinical benefit, we'd certainly want to go on to then establish if we could block onset of disease in healthy individuals who've been accidentally exposed to prions and infected, or those carrying one of the many mutations in the prion protein gene that we know will lead most of them to develop prion disease at some stage during their adult life. We'd hope it may be possible to prevent clinical onset by periodic treatment with PRN100. Now, I'll finish there, acknowledging very many people that have been involved in this work over the years, particularly the clinical team here at the National Prion Clinic. Simon Mead, many of you will know, who leads our clinical research team, and Peter Rudge, also another senior neurologist involved in the treatment of these patients. Our clinical fellows, Akin, Mark and Hans, and our wonderful nurse specialists, Kirsty, Salam and Veronica, were all key to this work. There are many other people I should really thank. I don't have time to go through them all, but they are, I hope, all listed in the Lancet Neurology paper. But again, Simon Mead was key to this, as was Azzy Khalili, who led the laboratory assays that were essential to underpin this work. And Caroline Potter, our business director, was essential in all the regulatory work that needed to be done to develop the drug product. I'd like to thank British neurologists who referred us patients and our colleagues at the Edinburgh Surveillance Unit. The Oversight Committee, I won't go into them in any detail, but particularly would like to thank the patient advocates on that committee, Colin Beattie, Peter Mills, and Charlotte Sain, who are really wonderfully supportive. It's so important, I think, to have patient advocates on these committees. And the Cure CJD campaign in the UK raised much of the funds required to do this work. And we're, of course, enormously grateful to them, as we are, of course, to the patients and families themselves that participated in this work. So the funders were the Medical Research Council, the National Institute of Health Research in the UK, the John Moulton Charitable Foundation, and the Cure CJD campaign. So I'll finish there, and thank you very much for your attention.
Well, thank you very much, Professor Collinge, for presenting your work. If I could just briefly summarize what I think you just presented. Very difficult to interpret such a small sample size. Like you said in one of your slides, basically you would have to see complete halting or improvement of the disease. But you believe, and your team believes, that there are some signals that suggest that you may see a treatment effect with PRN100. Is that a fair summation?
Yes, that's right. With these sort of numbers and doing the treatment in this way, that would be the only way we could conclude that the drug was working clinically. But I think the neuropathology we saw was clearly indicating that the drug was engaging with target and affecting the clearance of prions. So I think as far as we could conclude anything from this work, we were very encouraged by it.
And we do have some questions from people that are watching live. For those who are still watching live, if you want to submit a question, I would be happy to get to it if there's enough time. One question we had was about the normal function of PRPC, or the normal cellular prion protein. It seems that the purpose of it is not entirely well understood. Can you talk a little bit about that, and then whether or not developers of PRN100 or anything targeting that protein should be concerned?
Well, the protein doesn't appear to have a sort of simple unitary function. Various functions have been ascribed to it, and it's largely present at the synapse, the places where brain cells, neurons communicate with one another, and it seems to be involved in some adhesion and signaling processes at the synapse. But it's a part of a network of of proteins that are involved in complex functions there. So it's hard to ascribe to it a simple unitary function. But it does appear to have some redundancy in that you can, as I mentioned, engineer mice which completely lack the protein, and they seem to be pretty much completely healthy. So, one can survive without the prion protein. The key experiment, as I tried to illustrate at the beginning of my talk, was not just, because if you take a gene out, a protein out of a mouse when it's just a fertilized egg, an early embryo, adaptation might occur during neurodevelopment, which masks the function of the protein. But if you switch off expression of the protein in an adult brain, as the work I showed you we published nearly 20 years ago now in Science, that can be much more revealing in that the brain no longer has the capacity to have those sort of adaptations. And we found no problems in doing that. So it did give us a lot of reassurance that you can target the prion protein in the adult nervous system without severe consequences. Of course, that's in a mouse, and we can't be absolutely sure that's the same situation in humans. But this is the ideal target because we know that all infectious and all toxic prion protein species are made of the prion protein by definition. So if you restrict the access of that protein by blocking it with an antibody, it should stop all those downstream processes. So, as we've demonstrated, albeit only in 6 patients, but we saw no evidence of toxicity either acutely when we first infused the drug or in up to 8 months of treatment in one patient. We saw no evidence of toxicity. So far, it's been very encouraging from the point of view that it is safe to target this protein.
You touched on this a little bit at the very beginning, or actually at the very end. I think you made a comment that if these studies were positive, then there could be a consideration for perhaps using it prophylactically in people who carry a mutation for CJD who haven't yet developed symptoms. Of course, you treated many of these people when symptoms have already evolved. And as you said, it took 47 days for most people to have a treatment dose. Can you talk a little bit more about the potential of treating people much earlier, even maybe before they develop symptoms?
Yeah, so I think the next step is to do an efficacy study again in patients with CJD, but at a much earlier stage and with a much faster ramping up of the dosing. If that shows clear evidence of efficacy clinically, and it continues to be safe, we continue not to have any safety signals, then I think it is reasonable to consider whether you could then give it to healthy individuals who we know would develop the disease, particularly carrying certain pathogenic mutations in the prion protein gene that we know will lead to the onset of disease at some stage during their adult life. And there are now emerging biomarkers in blood and cerebrospinal fluid that can give us an indication of when the disease process is actually starting. Although the patients and their physicians may be unaware of it at that point, but we can see markers that the disease process has actually started. Then again, provided we have sufficient safety information, it may be worth considering offering that drug. Obviously, this would depend as on safety and regulatory approval. But in principle, it may be possible to prevent the disease starting or expressing itself clinically altogether. That would certainly be what we'd aim to do.
We get this talk or this question on all our talks, and that is, what about current treatment trials? And we were talking a little bit about this earlier before the presentation started, but we're both not currently aware of any current treatment trial. Of course, a drug company named Ionis released a statement last year at the last CJD Foundation virtual family conference that you can find there, and been in contact with that company, and things seem to still be on track for hopefully starting by the end of the year. PRN100, will we see future studies of that? And if so, when and what would that look like, do you think?
Well, we would, as I mentioned, like to do a formal efficacy study. What I just presented was, of course, not a research study. It was a treatment offered on a special needs basis to 6 patients. That was all the drug that we had. But what we'd like to do next is, and we've learned an awful lot, of course, about the dosing regime and safety issues now. So the aim would be to treat probably of the order of 50 patients with CJD, diagnosed at the earliest possible stage, and compare their rates of progression on drug with our large historical control patient collection from the National Prion Monitoring Cohort. That, I think, is the next step, and see if we can have clear evidence of clinical efficacy and hopefully continued safety. Now, when that's going to be, I'm getting this question a lot, as you can imagine, from around the world, understandably. We're in discussions with a number of potential funding partners at the moment to fund that study. This is going to be much more expensive than what I've just described. And it's a case of putting together the support to do that, which is what we're working on at the moment.
This may be an unfair question since you're still working on the support, but when PRN100 was ongoing, many patients contacted US clinicians and the CJD Foundation about getting access to the study and the study drug. Do you have any idea if any further study with PRN100 would be UK specific, or could it potentially be available to people outside of the UK?
Well, obviously the aim is that it will be available worldwide eventually, but our thinking at the moment is to do a study in the UK, in that there are probably sufficient patients in the UK to do the sort of numbers I'm talking about over a 1 to 2 year period. But we're certainly very open to discussion and have been in discussion with a number of colleagues in the world who want to join forces on that. And I think moving on to doing international studies, we'd certainly welcome.
Another question that we got, in treatments that are targeting PRPC, or the normal cellular prion protein, the idea is to stop the progression of propagation. What is the possibility that that type of treatment target could reverse the progression of clinical symptoms of CJD?
Well, I think if prion propagation stopped early enough, that should work. And, we saw that in the mouse model that I described. So I think the key is getting the treatment to the right level in the brain tissue before irreversible damage has occurred. Obviously, when this disease causes death of brain cells that have died, that there's no drug that's going to reverse that. And also, as I mentioned, when a great deal of cell death is occurring in the later stages of the disease, toxic substances are being released from degenerating cells, which in turn affects the cells around them. And that process, is not directly caused by the prions. It's a secondary process. And so I think you have to get there before that happens, because that is probably irreversible, at least with the sort of treatment strategy we have at the moment. So I think the key is to get there early, ideally, at the very, very earliest stages when CJD is first being considered as a diagnosis, and as ultimately going on to treat patients or individuals who are gene carriers who we know will develop the disease at some stage. So I think early diagnosis is going to be key to that. But all the science is suggesting that if we can get there early, we ought to be able to stop progression if we get there before too much cell death has occurred.
When you had mentioned 47 days that someone took to reach the treatment target or treatment dose, was that 47 days from diagnosis or 47 days from initial symptoms?
No, 47 days from us giving the initial dose. We started with a very low dose of the drug because obviously this was the first time this drug had been administered to humans ever. So, there may have been significant serious side effects. There could have been fatal side effects for all we knew. So we proceeded very cautiously indeed and started with a very low dose. Waited several days. The oversight committee reviewed all the data, and they gave us permission then to proceed to the next dose, and then after another few days working up to the highest dose that we gave, and then we repeated that dose at 2-week intervals. That dosing regime was a sort of guesstimate based on our previous animal work. We had no idea what to expect for sure in humans, but that was what came out, that it took 47 days on average to reach that target CSF concentration of 50 nanomolar. So now that we know that, and that we know, we haven't seen any adverse reactions, I think it would be reasonable now in a larger efficacy study to increase the dose much more rapidly and aim to get up to that target level very quickly, rather than do it in a cautious way that we inevitably did in this first treatment.
Well, just to put that in a little bit of context, so people kind of can understand where these patients were when they were treated. If it's 47 days from the first dose to the ideal concentration, what would be the mean time from onset or diagnosis until your first dose in most of these patients that you saw?
Well, it's very variable. I mean, I don't have the numbers in my head. It's all there in the paper if you want to look at those individual numbers. But I think the answer is it was too long. We want to get there earlier, but we moved as rapidly as we could. Of course, as I mentioned, with patients that lacked mental capacity, a court decision was necessary to authorize the treatment. The clinical team moved as rapidly as we could as soon as we'd identified a suitable patient that wanted to go ahead with the treatment. But I think undoubtedly we've learned a great deal from these first 6 treatments, and I think are confident that we can accelerate things considerably in a subsequent efficacy study. Of course, I think we'll need the cooperation, which I'm sure we'll have, of neurologists around the UK in terms of not waiting until they're absolutely sure about the diagnosis, for example, before referring the patient, and just as soon as the CJD is on the differential diagnosis, they think this is a possibility, just getting on the phone to us in London. And the National Prion Clinic has 3 mobile teams that go out to see people around the UK. And I think if we can get the funding to do an efficacy study, we'd want to probably have a number of regional centres around the UK that could recruit and treat the patients for convenience, so that we can do this, really very urgently when the possible diagnosis of CJD was considered. As you know, Brian, in most cases, it's not difficult to make a diagnosis in confidence in sporadic CJD with the right neuroimaging, and now with RT-Quick on CSF, one can, in most patients, make a very confident diagnosis early on, in specialist centres. So I think, that's what we want to achieve.
I certainly think it's reasonable to get people at an earlier stage, especially nowadays with improved diagnostics. But you just mentioned all the challenges that come to treating people with this illness as early as possible. It's definitely a challenge.
No, that's for sure, but we've got to meet that challenge, I think.
Definitely, we want to see treatment. Some of us who have been following your talks over the years, we've seen the development of PRN100 and your search for finding funding, and now finally the results of the treatment program. Have there been any further research or update onto the PRNP antibodies themselves? Are we sticking with the current antibody, you think, for any future studies?
We're sticking with the current antibody. we may possibly modify it in future studies. I mean, there are new technologies emerging to modify antibodies so that they cross the blood-brain barrier more efficiently. And that's something we've been thinking of. Of course, that requires yet further funding and partnerships to do that. But we are in discussions about possibly modifying the antibody to make it so it gets into the brain more efficiently. We're getting enough in, we think, with the current treatment protocol, but it's a lot of antibody we have to give to do that, which would ultimately be very expensive. And so if we could modify the antibody in a way that made, say, 20-fold more efficient crossing the blood-brain barrier, you know, the dose could be correspondingly smaller and more patients could be treated. So that's something we're considering. But this antibody, you know, binds to the human prion protein with extremely high affinity. And, you know, we have human data on it now. These things, as you know, are not easy to do. And so we're keen to move forward with the antibody that we have. But as I say, we may consider some modifications to make it access the brain more efficiently.
I believe patients in this study were dosed every 2 weeks. Is that correct?
That's right, yeah.
Do you foresee any lengthening of dosing periods? Are people going to need to receive doses for the rest of their life if this is a successful treatment? How do you envision that working?
Well, the 2-week treatment, again, was a sort of educated guess. You know, we did measure as we describe in the paper, we did measure the half-life in serum after termination of treatment. And probably the dosing could be extended to some extent. The intervals could be broadened to some extent. As you saw from the rise in CSF levels, you know, it was still going up, you know, and that probably one doesn't necessarily need to treat every 2 weeks. The aim by you know, the prions are not indestructible. You know, the brain turns over prions. You know, you can do these experiments also in mice. We and others have done this, where you can follow how rapidly brains can clear prions, and it's really quite efficient. You know, the half-life in our experiments was about 2 or 3 days. So, you know, if you block prion propagation, at least in a mouse brain for a month or so, you should cure the infection. And that would be our aim in sporadic CJD, to actually eradicate the infection. So we wouldn't hopefully need to be offering this treatment lifelong. It would be a course of treatment that would, we hope, eradicate the infection, or at least knock it down to levels that were not a problem, and then treatment may need to be given much less frequently if there was any recurrence. This is all to learn going forward. We can't predict the answers to these. With people carrying gene mutations, where, you know, our dream would be to prevent onset of the disease at all, I don't think one will be talking about very frequent treatments there. Again, that would only come from experience, but it may be that people would only need to come in for an annual treatment with PRN100 to knock down any prion propagation that was occurring, and then they would come in 6 or 12 months later for various investigations to see whether there was evidence, looking at biomarkers to see if the disease had started up again, and if so, another course of treatment. If not, then, you know, give the good news and see them again in 6 or 12 months' time. That's what we hope would be the case with the inherited forms. Of course, with the inherited forms, the mutation is there, and prion propagation will presumably start again even after you've dealt with the initial problem. So one would anticipate it would be lifelong treatment, but perhaps only on an annual basis. That would be my hope. But again, all this is for the future. We can only find out these things for sure by actually doing it.
Unfortunately, it's been very difficult for you to secure funding to do an appropriate clinical trial. Could you give people who are not familiar with clinical trials just a ballpark estimate of how much funding you would need to do a proper clinical trial for prion disease?
It depends on quite a few variables, and one of the major ones here is producing enough of the drug. Monoclonal antibodies are expensive to produce, and particularly when it's not an established manufacturing process. These are bespoke productions, which are intrinsically expensive. So you're certainly talking about some millions of dollars just to make the drug product we'd need to do an efficacy study now. You're probably talking, of the order of $10 million to do the study I'm talking about. So it's, in pharmaceutical terms, it's not a huge amount of money. For us as academics, this is a lot of money. But it is very frustrating when you see, in comparison, the money that's spent on Alzheimer's trials, for example. I feel, can't you just give a small percentage of that to us to work on CJD? But it has proved a struggle. Of course, this is a rare disease, and there isn't a traditional business case here. The patient charities in the UK have been wonderful and still are wonderful raising funds, but not at that level. But that's the sort of ballpark we'd be looking for to do, and that's in an academic environment. It would cost much more that, I think, than that it was done by a pharmaceutical company. But our hospital is extremely supportive, and I think would provide facilities and so on. And obviously, there's my MRC unit that has all its facilities. So we think we could put that sort of study together for about $10 million.
So a related question was why doses were only made for 6 patients. I'm assuming that's partly because of the cost, but could you talk a little bit about what's involved in making a monoclonal antibody? Why is it so costly? What's that process like?
Yes, so we didn't make it for 6 patients. We simply made a first batch, and that was all the funding we had to make that first batch, which cost something over $1 million to make that first batch some years ago. How you make monoclonal antibodies is in cultured cells. So you have to genetically engineer particular cells so that they make the antibody for you. And these are grown in facilities in large tissue culture bags, hundreds of liters of fluid containing these cells, which grow up over a period of time, and then the cells are making the antibody, and then you have to extract and purify the antibody from that culture medium, concentrate it, and prepare it for human use. Those steps are not things we can do. They have to be done by a manufacturer that has the appropriate regulatory approval to manufacture products for human use, which is inherently very expensive. So monoclonal antibody drugs and lots of the latest drugs, particularly being used to treat cancer now, are monoclonal antibody drugs. They're all expensive because and part of that is the cost of manufacturing the drug itself.
This is a multi-part question, and it's referencing the possibility of an efficacy study where you'd have to enroll approximately 50 or so patients. The first part of the question is, wouldn't it mean a pretty long time period as only a fraction of those patients will be diagnosed correctly, and only a part of those would possibly participate in such a study? So I think that was kind of asking about your target population in the UK itself. And the second part of that question was, how would that kind of very early diagnosis be? Wouldn't you be at risk of including other rapid forms of dementia besides prion disease?
Well, we see about 120 new patients with sporadic CJD in the UK every year. In our experience so far, about a quarter of those we see at a sort of early stage that we're looking at. We want to look at here. So that would be about 30 patients a year. So we think it is tractable, but as I mentioned earlier, I think in partnership with neurologists across the UK, if they knew a trial was going on and that it was important to recruit patients early, I would hope and expect, they'd be referring patients to us much more rapidly rather than necessarily waiting until relevant tests were back. They'd say, we think this is CJD. Would you like to see this patient? And I think we could accelerate earlier diagnosis. Some of the forms of sporadic CJD, as you know well, Brian, are incredibly fast, and I think in those patients it would be a real challenge to get there early enough. The second part of the question is, might we make some diagnostic mistakes here by trying to get there too early? Well, I don't think so, because we would only treat if we were confident about the diagnosis. And I think if you have, the characteristic imaging changes, magnetic imaging changes, and diffusion weighted images that are very characteristic of CJD, and you had a positive RT-QuIC in the CSF, the diagnosis is virtually certain. And I think together with, of course, the clinical picture. And so I think it'd be highly unlikely we would make any errors with that. Of course, at the end of the day, it's up to the patients and their families whether they want to participate in this, and that's entirely up to them. But in principle, we think that sort of study is doable over perhaps a 2-year period. But certainly, as I've also mentioned, we'd be entirely open to international collaboration to increase numbers. There are regulatory complexities, as you know, in doing trials across international borders, but they're not insurmountable. So I think it can be done.
Related question, say there was a diagnostic error. Is there any danger in giving PRN100 to someone who may not actually have CJD?
Well, not that we've seen so far, but of course it's only based on treatment of 6 individuals. So it's a small amount of data, but if you do a traditional phase 1 safety data, it's usually on a relatively small number of individuals. So far we haven't seen any safety signals, and so there's no particular reason to suppose if we gave it to someone that didn't have CJD that would be any different. But as I've mentioned, I don't think we would give it to anyone who didn't have CJD, because I think we can be confident about the diagnosis with the tests that I've mentioned. If we weren't confident, then I think in the context of a clinical trial, we wouldn't give it. There would be strict inclusion criteria. They wouldn't be met.
Just wanted to end on a final question. That is, you've been in this field quite a long time and seen it grow quite a bit. What is your overall outlook regarding a potential treatment for this disease in our lifetime, do you think?
Well, I'm pretty optimistic. I mean, we were very excited, as I mentioned, by the changes in neuropathology we saw with the drug, the lack of toxicity and the levels we were reaching. And I can't guarantee anything, of course. We may just be being over-optimistic, but I think there's a reasonable possibility that getting there early with this, and there are other methods of targeting the prion protein itself, one of which you've already mentioned. But I think if we can target PRP effectively in the brain early enough, it should work. And we understand enough about these diseases now. I mean, we understand the biology of these diseases much better, in my opinion, than we understand the biology of Alzheimer's disease. That this is a clearly validated target. And if we can hit that target effectively enough, early enough in the brain, it should work. So I'm pretty confident, actually. But we'll follow it through. But we need to get the support and funding, I think, to do the next stages as ever.
Well, Professor Collinge, it is now 11 p.m. your time. Thank you very much for taking this evening to talk with us and the family members. We really appreciate the update that you gave us. For those that are watching, if you want to watch it again, it will be recorded and put on YouTube and the CJD Foundation website. Thank you very much. Let you have any final word if you have any.
No, I'm just delighted to have been able to speak to you. Thank you very much for inviting me, and best wishes to everyone.
Thank you.